Hello there, my name is Arne Huyst and I work in the pain group of Ghent University. In the PROFIBFUN project we investigated proteins that form amyloid fibrils. In the previous videos we talked about the presence of amyloid fibrils in food, how they look like and how we can promote their formation. The next thing we wanted to investigate was, can amyloid fibrils be used to prepare better emulsions? Well, let me first explain you what emulsions are. Emulsions are food systems such as butter, milk or mayonnaise. They have all in common that they are stable mixtures of two liquids, being water and oil, which are normally unmixable. However, by mixing them, the oil can be broken down into very small droplets which float in the water. Nevertheless, just mixing them will only keep these small droplets for a very short time before they merge again. To prolong this, we have to add another component which is called an emulsifier. So what are these emulsifiers? An emulsifier is a component which absorbs onto the oil droplets and makes it possible to keep them small for a longer time. In the food industry, proteins are often considered as good emulsifiers. As proteins are quite large and can interact with other proteins absorbed onto the oil droplets, they can form very strong layers around the oil droplets. This limits their merging and hence results in a better emulsion stability. Now, the question arises, if emulsions using proteins as emulsifiers are already good, why do we need to study protein fibrils? Of course, emulsion stability can always be further improved. Especially, proteins are often not good in stabilizing emulsions with a very high oil content, such as mayonnaise. Furthermore, most of the proteins which are good emulsifiers are animal-based, whereas most plant-based proteins are not well researched up till now. As such, protein fibrils can be helpful to improve the stability of food emulsions. Based on the insights obtained in the PROFIBFUN project, we studied the emulsifying properties of protein fibrils, from ovalbumin, which is the main protein present in egg whites from hen eggs. For food applications, we can either form fibrils by just heating ovalbumin, or by heating ovalbumin and hydrolyzing, so in fact cutting the resulting fibrils, with an enzyme. When the protein is only heated, mainly short fibrils are formed. In contrast, the additional enzyme treatment first broke down the structures and eventually those assembled into much larger fibrils, that were even so large that they entangled with each other and formed a gel structure. We then prepared emulsions containing a high oil content with both types of ovalbumin fibrils as well as ovalbumin without fibrils. The emulsions without fibrils showed large oil droplets, and the emulsions quickly separated an oil phase, showing a low emulsion stability. Similar emulsions with the short fibrils initially already contained much smaller oil droplets, and also the oil separation occurred to a much lower extent. Finally, the emulsions with the long fibrils contained even smaller droplets, and they did not show any oil separation at all. This clearly indicated that the large fibrils improved the emulsion stability, which was linked to their increased viscosity of the gel structure. So yes, protein fibrils are indeed beneficial for improving the emulsion stability, and it seems that their length has an impact as well. This is certainly interesting, considering the implementation of protein fibrils in food products. Of course, it is important to know what the effect is of these protein fibrils in the human body when foods containing these protein fibrils are eaten. Well, we will find that out in the next movie.